of Molecular Biology.
Kids, do you know who this is? He's a molecular biologist and a biophysicist and was the first scientist to conceptualize central dogma of molecular biology in 1957. It explains how RNA is created from DNA, the human or deoxyribonucleic acid is subsequently translated into proteins, which are made by the sequence of amino acids DNA to RNA to proteins. Yes, this process is vital for all living things, plants, animals. Humans need their own protein to function well. It shows a deeper understanding of how genes work.
I think I've been eating a lot of junk food this past few days. What can I eat instead to stay healthy? Oh, an egg! Eggs are a good source of protein. It makes you lose weight, gain muscles, mass, and lower blood pressure. But how do we produce proteins?
In central dogma of molecular biology, there are three processes. First is the DNA replication, wherein it duplicates the genetic information into a full copy of DNA. Second is the transcription, where DNA transcribed to RNA, a single-stranded polymerase, and an enzyme that reads a particular region in DNA, and serve as a messenger from molecules to nucleotides. Last step is the translation between RNA and proteins. After transcribing, amino acids are being transported from molecules to ribosomes, which is the biological machinery that makes protein in cytoplasm. The tRNA anticodons corresponds to codons or triplets of nucleotides on the mRNA following the instructions to link the amino acids in the correct order to make a protein.
Proteins are the basic building blocks of life. So kids, don't forget to eat more eggs for more proteins in our body and be healthy. Oh, where are my eggs?
Whoa, I'm so full. I think those eggs are too heavy. Too bad, I have a slow metabolism. Oh, hi kids! Don't worry, not all humans have slow metabolism. Some have a fast metabolism because of their enzymes, which help them have a fast metabolism and break down substances in their bodies. Speaking of enzymes, there are enzymes in the concept of central dogma of molecular biology.
Enzymes plays a role in the one gene, one enzyme hypothesis, which suggests that each gene encodes a single enzyme.
This is Archibald Garrod, the one that proposed the role of enzymes, and this idea was mutually confirmed by Beadle and Tatum through genetic and biochemical studies of the bread mold Neurospora. Enzymes occur during transcription and translation. Steps enzymes hypothesis shows the importance between the connection of genes and enzymes. During the process of replication, transcription, and translation, some enzymes are included, namely DNA polymerase, DNA ligase, and reverse transcriptase.
Excuse me, Nash. Have you seen Jade's egg? She's hungry.
Oh no, I have not noticed anything.
What are you doing?
I'm explaining to kids what the enzymes present in molecular biology.
Perfect, I know that. Kids, come over here. This picture is a DNA polymerase. You see, there are two DNA polymerase. First, one structure. This structure synthesizes DNA from a DNA template used to radio-labeled DNA probes, polymerase, chain reaction, and cDNA cloning. Next over here is the DNA ligase, which is assigned to create a DNA cloning by forming the single strand ends of a DNA to a covalent bond, which occurs during replication, which is the DNA cloning. Lastly, this image shows reverse transcriptase. There is also a step called reverse transcription, which is as important as the normal transcription step, as it synthesizes DNA from a RNA template, and important for DNA or cDNA cloning, which are the copies of RNAs.
Did you listen well to them, kids? You may ask why we need to study and learn about the central dogma of molecular biology.
Well, let me explain to you why we need to study it and why is it important based on its functions. The first function of the basic concept of molecular biology is that it states the DNA codes for RNA while RNA codes for protein. It has the blueprint needed to assemble the RNA, the proteins that make up the body structures, and perform the majority of its tasks. Second, it serves as a book of knowledge on how genetic information processes works, from DNA to RNA to proteins, elucidating the key processes involved in gene expression and the foundation for molecular biology as a scientific discipline. Third, of course, the central dogma of molecular biology is a crucial process and is indeed important as the processes included here are fundamental of the functioning of living organisms.